Dear students, today we will discuss nanoparticles and biological system. In this lecture, we will discuss a bit introduction that how nanoparticles are involved in biological systems. In previous lectures, we have discussed nanoscience, then nanotechnology. That I have discussed that these are basically the dimensions between approximately 1 and 100 nanometer are basically known as the nanoparticles, and first time this term was coined by Norai Taniguchi, particles between of Tokyo Science University, to describe the semiconductor processes. Previously we have discussed about its history and characterization, that we can characterize nanoparticles on the basis of origin, on the basis of dispersion, on the basis of shape and structure, chemical composition, aggregation state, and surface modifications. And on the basis of origin, we have described them into natural processes, that there could be lightning which is involved in nanoparticle synthesis, then there could be thunderstorm, volcanic eruption that leads to nanoparticles formation. Second origin could be unintentional release, for example any unattended activity that leads to nanoparticles formation. And third origin, mode of origin, is manufactured, that we intentionally synthesize nanoparticles using physical method, chemical method, or any kind of biological method. Same if we go about dispersion, that could be because of gases, for example aerosols, in case of liquids, gels, ferrofluids, and that could be dispersion could be in solid state, for example matrix materials. Similarly, we can characterize nanoparticles on the basis of shape and their structure. For example, they could be spherical, they could be needle-like, and they could be in tubes. Similarly, we can characterize nanoparticles on the basis of their chemical composition. For example, they could be metals, they could be metal oxide, they could be polymers, they could be biomolecules. These could be semiconductors and compounds. Similarly, in case of aggregation state, they could be aggregates, they could be single particles, or they could be agglomerates. And similarly, there are some surface modifications which leads to nanoparticle synthesis. For example, they could be untreated, when there is just the nanoparticles in the way they have been synthesized. There are some modifications using the different kind of coatings, using conjugates, using polymer films. Similarly, some cores and shells are also formed on the nanoparticles to do their surface modifications. Now these cores could be spherical, or these could be capsules. This is the overall picture which is indicating the characterization of nanomaterials. As I have discussed in the start, that nanomaterials in biological systems, how you can see in this picture that there are some things of sizes of the things. For example, water is 10 raised to the power -1 nanometers. Similarly, glucose is 1 nanometer, antibody is 10 nanometers. These are some classifications which is indicating that the size, every particle, everything has a size. That could be nanopores, that could be dendrimers, that could be nanotubes or contouring dots. The greatest discovery was that there is chemistry which is involved in biology or in life sciences, and that greatest discovery was the discovery of double helix structure of DNA by Watson and Crick. According to this is the DNA structure that has the chemical composition. Here is the thymine, adenine, cytosine, and guanine. So hydrogen bonding is present between adenine and thymine, similarly guanine and cytosine, that base pairs in DNA, and there could be minor groups and there could be major groups, and this is all involved chemistry and biology. Lesson what you learn from nature, that DNA bound by base pairs, and there is beginning of unwinding of major events which are occurring at nanoscale. Similarly, some lipids and proteins, these are also at nanoscales. Self-assembly of the tobacco mosaic virus. This is basically the self-assembly of tobacco mosaic virus, and this is basically the viral DNA, RNA. So, how do we get to this level of complexity? This is because of programmed self-assembly. Here in this picture, you can see the hierarchical assembly of molecules to mammals. Here you can see this is the human being that are in macromolecular in structure. Then, if we take a small part which is 1 millimeter in nature, then we more magnify into 100 micrometer. This is the structure which is 5 micrometer in nature. Then is the 1 micrometer, 100, and there is the structure which is in nanometers. So everywhere in the life or biological assemblies or biological systems, there are some nanoparticles or nanomaterials which are involved or which are taking place. Everything which is in macro scale or in at least in a mammal or any living organism, there are the self-assembly or hierarchical assembly of molecules or nanoparticles into the mammals, which are leading to the synthesis or formation of a biological system. Thank you.